Hey everyone, if you're a medical coding student and you're ready to jump into one of the most fascinating and, yeah, kind of complex areas of the field, then you are in exactly the right place. Today we're going to build a solid foundation, a guide to CPT molecular pathology. Okay, let's get into it. So here's our game plan. First, we'll start with the big picture, just to get our bearings. Then, we're going to cover the single most important rule in all of molecular coding. After that, we'll dig into the nitty-gritty details. We'll learn some of the lingo so you can talk the talk. And finally, we'll map out what to do when you just can't find the code you're looking for. All right, first up, decoding molecular pathology. Before we get into the weeds, let's just zoom out and understand what we're actually coding for and, why it's so important. At its heart, molecular pathology is all about reading our body's own instruction manual. Our DNA and RNA. These tests are incredible. They help doctors diagnose, treat, and even monitor all kinds of diseases. Our job as coders is to take that super complex scientific analysis and translate it perfectly into the language of CPT codes. Okay, so you'll basically see two main buckets for these conditions. On one side, we have what are called germline conditions. Think of these as things you're born with, the genetic traits passed down from your family. Then, on the other side, you have somatic conditions. These are changes that happen after you're born, like the genetic mutations we often see in cancer. The codes we use have to reflect which type of test was done. Let's move on to section 2, and I really want you to pay close attention here because this is it. This is the absolute core principle of molecular coding. If you only remember one thing from our entire chat today, make it this. CPT code selection is all about the gene. It is not about the patient's symptoms or what disease the doctor thinks they might have. It all comes down to one question. Which specific gene or genes did the lab analyze? And you'll notice the CPT manual uses the official gene names, and if you see any diseases listed in a code description, remember, those are just examples, not a complete list. This right here is the takeaway. Let this really sink in. It does not matter if a doctor ordered a test because they suspected breast cancer or colon cancer. As a coder, your job isn't to worry about the why. Your only question is, what exact gene did the lab test? The why is for the doctor. The what is for you. Okay, on to section 3. So now that we have that fundamental rule totally locked in, let's look at some of the common nuances you're going to run into all the time. This is where you go from being a good coder to a great one. This is a big one. Some lab tests are just looking for a few specific known variants within a gene. Think of it like looking for a couple of known typos on a page in a book. Other tests, though, do full gene sequencing, where they read the entire page from top to bottom. A code for targeted variants only covers the specific ones listed in its description. You can't use that code for a full sequence. It's a totally different test. Have you ever wondered what's actually packed into a single molecular pathology code? Well, it's a lot. It's basically the whole shebang. As you can see here, one code typically bundles the entire lab process, from breaking open the cells to get the DNA, to extracting it, prepping it, and finally detecting the gene they were looking for. It really is a full package deal. And hey, a quick but really important note about modifiers. If a provider is only interpreting the results of a test that somebody else performed and then writing up the report, you're going to want to append modifier 26. This just separates the doctor's brain work from the technical lab work. It's simple, but it's absolutely critical for getting it right. All right, let's switch gears a bit for section 4. To code this stuff accurately, you've really got to speak the language. So we're going to do a quick vocab round. Knowing these terms is going to make you feel way more confident when you're reading those lab reports. First up, allele. This sounds complicated, but it's not. If a gene is like a recipe for eye color, the different alleles are the specific instructions for blue eyes or brown eyes or green eyes. It's just a variation of a gene. Simple as that. Next, we have copy number variant, or CNV. Now, this isn't just a tiny change in a gene. A CNV is when a whole large chunk of DNA is either missing completely or it's been duplicated. So you either have too much of that genetic information or too little. It's a big picture change. So this is key. Not all genetic variants are created equal. You'll hear the term mutation, which means a variant that's pathogenic or known to cause disease. But then you have polymorphism. That's just a common, harmless variant that isn't linked to disease. Knowing the difference is huge when you're trying to understand a report. And that brings us to our final section. We've gone over the rules and the lingo, but what happens when you follow all the steps and you still come up empty-handed? Don't worry, this section gives you a clear step-by-step plan. It's a situation every single coder faces sooner or later. You've got the lab report, you know exactly which gene was tested, but you're flipping through the CPT manual and nothing. It's just not there. The good news is you don't have to panic. There is a standard process for exactly this scenario.
Here it is. This is your decision tree. Step 1, you always search the Tier 1 codes first. These are for the most common gene tests. If you strike out there, you move on to Step 2. Check the Tier 2 codes, which cover less common genes. And if it's still not there, then and only then do you use the unlisted procedure code, 81479. It's a logical 3-step process every time.
So there you have it, the essential guidelines for finding your way through molecular pathology coding. We covered the big picture, we learned the golden rule, that the gene is always the key, and we have a clear path to follow even when things get tricky. You now have the tools. You've got the decoder's key so you can approach those lab reports with confidence and tell their stories accurately.